After studying this module, you shall be able to know about ultrasound reaction, learn about cavitation, identify how ultrasound is useful in organic chemistry, evaluate the factors affecting the ultrasound, and lastly, analyze Refimitsky reaction, Grignard reaction, cyclopropanation and Ullmann reaction, etc. Let us start by the introduction of this topic. Ultrasound is simply sound pitched above human hearing. It has found many uses in many areas. At home, we use ultrasound for dog whistles, alarms and jewelry cleaners. In hospitals, doctors use ultrasound to remove kidney stones, to treat cartilage injuries and to image fetal development during pregnancy. In industry, ultrasound is important for emulsifying cosmetics and foods, welding plastics, cutting alloys and large-scale cleaning. None of these applications, however, take advantage of the effects that ultrasound can have on chemical reactivity. The first report about the effect of ultrasound on chemical reactions was reported by Richards and Looms in 1927, involving rate studies on the hydrolysis of dimethyl sulfate and the iodine clock reaction, that is reduction of potassium iodate by sulfurous acid. Whereas the first commercial application of ultrasonics appeared in 1917 when Langevin's echo sound technique for the estimation of depths of water resulting in the system known as sonar, that is sound navigation and ranging. Then in 1938, Porter and Young reported that ultrasound increased the rate of the Curtius rearrangement. In 1950, Renaud prepared an organometallic compound using ultrasound. Hahn and Bajok in 1982 reported significant increase in yields and rates of Refimitsky reactions. Now let us start by talking about ultrasound. The term ultrasound refers to sound waves ranging in frequency from 20 kHz to 10 MHz. The chemical effect observed during sonification, that is high intensity ultrasound waves, is acoustic cavitation. Cavitation is the formation of collapse of microbubbles produced in a solvent medium while irradiating with ultrasonic waves. When ultrasonic waves are passed through a medium, the particles experience oscillation that leads to regions of compression and rarefaction. The negative pressure in the rarefaction region gives rise to the formation of bubbles that may either filled with a gas or the vapors of the liquid, or may be almost empty depending upon the pressure and the forces holding the liquid water. The use of sonification has been widely applied to heterogeneous systems, which generally activates the solid present. For homogeneous system, aqueous solutions have been the focus of investigation because for many years it has been wrongly assumed that only water dissociates into H and OH radicals, which go on to carry out a variety of reactions. The major sonochemistry resulting from cavitation in homogeneous solutions is accepted to be generated during the collapse of a bubble, producing extremely high temperature and pressure. The temperature and pressure so produced have been estimated to be of the order of 5200 degree Kelvin and 1 kilobar. The solvent plus any volatile solute which have diffused into the sonification bubble therefore can undergo reaction due to the direct action of cavitation. The liquid surrounding the collapsing bubble has also been speculated to be a reaction zone. Ultrasound can be generated in a number of ways. However, the most common techniques are the use of an ultrasonic transducer. This is a device which contains a piezoelectric material which is usually a ceramic. When voltage is applied to the piezoelectric element, the electric energy is converted to a mechanical vibration. This in turn produces a sound wave. By approximate choice of the element, the frequency of the sound wave can be varied whether in the audible or ultrasound range. At least 5 different types of sonicators are used in chemical reactions. The simple ultrasonic cleaning bath is by far the cheapest source of ultrasonic irradiation for the chemical laboratory. As you can see in the figure of the ultrasonic cleaning bath. It's a bath which contains water and detergent attached to transducers and has the reaction mixture placed in it. This is the most accessible method because of the ready availability of commercial equipment. The ultrasound is produced by the transducers mounted on the external walls of the bath. These usually operate at a power density of 1 to 5 tungsten per centimeter square. The ultrasound is transmitted through the fluid medium, usually H2O, and into the reaction solution contained in the flask. Addition of detergent reduces the surface tension, that is, lowers the energy required to pull apart the water molecules, and the disturbance in water is greatly increased. Now let us talk about cavitation. Sound, including ultrasound, is transmitted through any physical medium by waves that compress and stretch the molecular spacings of the medium through which it passes. As you can see in this figure, which shows the creation of stable cavitation bubbles and creation and collapse of transient and stable cavitation bubbles. Part A depicts displacement, B shows transient cavitation, C shows a stable cavitation, and D depicts the pressure. As the ultrasound crosses the medium, the average distance between the molecules will vary as they oscillate about their mean position. When the negative pressure caused for an ultrasonic wave crossing a liquid is large enough, the distance between the molecules of the liquid exceeds the minimum molecular distance required to hold the liquid intact, and then the liquid breaks down and voids are created. These voids are the so-called cavitation bubbles. As the liquid compresses and stretches, the cavitation bubbles can behave in two ways. In the first, called stable cavitation, bubbles formed at fairly low ultrasonic intensities oscillate about some equilibrium size for many acoustic cycles. In the second, called transient cavitation, bubbles are formed using sound intensities in excess of tungsten per centimeter square. As you can see in the diagram, transient cavitation and the origin of sonochemistry, where you can see the expansion and the compression, the acoustic pressure, then the implosion and shock wave, and we have the hot spot and rapid quenching. So this figure shows bubble size and cavitation dynamics. Liquids irradiated with ultrasound can produce bubbles. These bubbles oscillate, growing a little more during the expansion phase of the sound wave than they shrink during the compression phase. Under the proper conditions, these bubbles can undergo a violent collapse, which generates very high pressure and temperatures. This process is called cavitation. Transient bubbles expand through a few acoustic cycles to a radius of at least twice their initial size, before collapsing violently on compression. Transient bubble collapsing is considered to be the main source of the chemical and mechanical effects of ultrasonic energy. Each collapsing bubble can be considered as a microreactor, in which temperatures of several thousand degrees and pressures higher than 1000 atmosphere are created instantaneously. From an analytical chemistry point of view, most of the effects of interest regarding ultrasonification are related to cavitation. Cavitation causes solute thermolysis along with the formation of highly reactive radicals and reagents such as OH radical and H2O2, which induce drastic reactive conditions in the liquid media. In addition, if a solid is present in solution, the sample size of the particles is diminished by solid disruption, thereby increasing the total solid surface in contact with the solvent. In this way, ultrasonification remains unique, since no other method of sample treatment can produce such effects. Generally, ultrasonification aids chemical analysis by 1. Enhancing solid-liquid elemental extraction, 2. Shortening sequential extraction schemes for elemental determination, 3. Speeding up solid-liquid extraction of organic species and enzymatic reactions, 4. Accelerating electroanalytical measurements by enhancing mass transport efficiency. 5. Accelerating liquid-liquid extraction techniques. 6. Enhancing the performance in solid phase extraction and microextraction. And 7. Incrementing accuracy in the solid matrix dispersion technique. Next, let us study about the ultrasound sources. A variety of devices have been used for ultrasonic irradiation of solutions. For common laboratory use, there are three main designs, namely ultrasonic cleaning bath, cup horn sonicator, and direct immersion ultrasonic horn. The source of the ultrasound is a piezoelectric material made of lead zirconate titanate ceramic PZT or quartz, which is subjected to high voltage alternating current with an ultrasonic frequency ranging between 15 kHz to 10 MHz. The piezoelectric material expands and contracts in this electric field and is attached to the walls of the cleaning bath, which converts electrical energy into sound energy. As you can see the figure, which is showing the apparatus for carrying out a reaction in an ultrasonic cleaning bath. In cleaning baths, the acoustic field is continuous, whereas it is in the pulsed form of probes. In the ultrasonic cleaning bath, a liquid is present for transferring the ultrasound from the generators to the reaction vessel. The vessel on sonification is adjusted to the point where cavitation, that is bubbles, is maximum. The temperature of the water is kept constant by cooling. Results obtained by using a cleaning bath are not always reproducible, and therefore at times the use of sonic probes is preferred. Next, we have a figure showing us the Susslich cell apparatus attached to a sonic probe for use in small scale reactions in an inert atmosphere. Next, let us talk about ultrasound in organic chemistry. Ultrasound can be used in organic reactions since it provides specific activation based on a physical phenomena called as acoustic cavitation. The liquids irradiated with high intensity ultrasound acoustic cavitation, that is, the formation, growth and collapse of bubbles, provides the primary mechanism for sonochemical effects. In cavitation, bubble collapse produces intense local heating, high pressure and very short lifetimes. These transient, localized, hotspot drive high energy chemical reactions. It is well established fact that these hotspots have temperatures of above 5000 degrees Celsius, pressure at around 1000 atmospheres and heating and cooling rate above 1010 Kelvin per second. Thus, cavitation serves as a means of concentrating the diffuse energy of sound into a unique set of conditions to produce unusual materials from dissolved and generally volatile precursors. As far as solids or solid gas systems are concerned, chemical reactions are generally seen in the ultrasonic irradiation. In addition, the interfacial region around cavitation bubbles has very large temperature, pressure and possibly electric field gradient. Liquid motion in this vicinity also generates very large shear and strain gradients. These are caused by the very rapid streaming of solvent molecules around the cavitation bubble, as well as the intense shock waves emanated on collapse. There are various applications of ultrasound in organic chemistry, namely homogeneous ultrasound chemistry in aqueous medium and in non-aqueous medium, heterogeneous ultrasound chemistry. We have three categories: phase transfer catalysis, reaction with metals and heterogeneous catalysis. Number 3, enzyme reactions pro-offered in ultrasound. For example, sonolysis of tetracarbonyl ion in decane under argon atmosphere produces amorphous ion upon decarbonylation instead of crystalline ion, which shows that both very high temperatures and also rapid cooling rates, that is of the order of 106 kelvins per second, are involved. The more volatile pentane yields Fe3CO12, indicating a somewhat slower collapse. It has been estimated and calculated that the pressure within a bubble in water can rise up to 2000 atmospheres and the temperature can reach up to 5000 degrees Celsius during a collapse. As these bubbles are small and they rapidly collapse, they can be seen as microreactors that offer the opportunity of speeding up certain reactions and also allow mechanistically novel reactions to take place in an absolutely safe manner. Many reactions can be conducted even in a simple ultrasonic cleaning bath, although the amount of energy that reaches the reaction is only between 1 to 5 tungsten per centimeter square, and temperature control is normally poor. Large-scale reactions can be better conducted using immiscible ultrasonic probes that circumvent the transfer of the energy through water and the reaction vessel. These are two types of effects mediated by ultrasound, the chemical and physical. When the quantity of bubbles is low, it is mainly physical rate acceleration that plays a role. For example, a specific effect is the asymmetric collapse near a solid surface, which forms microjets. This effect is the reason why ultrasound is very effective in cleaning and is also responsible for rate acceleration in multiphasic reactions, since surface cleaning and erosion lead to improved mass transport. Next, let us talk about reactions accelerated by ultrasound. A number of common reactions used in synthetic organic chemistry can be carried out efficiently using ultrasound. There are several advantages. Generally, with the increase in yield, the percentage of byproducts decreases. Reactions occur faster so that lower temperatures can be used. Ultrasound provides alternative pathways for reactions due to the formation of high energy intermediates. Ultrasound also enables reactions involving organometallic reagents to be carried out safely. In the following text, several specific applications are provided. Let us just have a look at them. Organometallic reactions is what we have next. We start with Refaminski reaction. Now you can see in the given equation, when using ultrasound at room temperature for 5 minutes, the yield is 95% as compared to the conventional method of stirring at 80 degrees Celsius for 12 hours, which gave a much lower yield of 61%. Next, let us look at the example of Siemens-Smith cyclopropionation carried out in presence of ultrasounds using zinc and diiodomethane. The yields obtained were extremely high, 99%. After this, we have some condensation reactions listed here. The first one being ultrasound on a heterogeneous reaction. We have a table, a small table, which is analyzing the products obtained. We have A, B, C and D, where A has H as the substituent, B has methyl, C has methoxy and D has NO2. The two products numbered 7 and 8 are shown in the diagram. The first column is for structure 7, the second column is for structure 8, and the next column again for structure 7 and then for structure 8 respectively. It is showing the ratio of various isomers formed, where it is changing from 0 100 to a 100 0. 55 45 is changing to 93 7. 45 55 is changing to 94 6, and a 0 100 is changing to a 20 80, where the conditions are changed from the conventional method using ethanol as the solvent and refluxing to the use of ultrasound. In our next example, we have the equation showing the synthesis of 3,4-dihydropyrimidine 2,1H-one. Using the ultrasound with methanol as the solvent, the yield obtained for this reaction was as high as 92%, whereas when carried out the same reaction with the same products, when carried out without ultrasound, gave us a very average yield of 60%. After this, now let us have a look at cycloaddition reactions. The first one, let us talk about Diels-Alder reaction. Leck and Cinder observed that cycloaddition between vinyl cyclohexene and the dienophile leading to a duct is 65% yield under use of ultrasound. Thus, it improves the regioselectivity among the two regioisomers obtained predominantly. We have a small table listing the conditions when using ultrasound. The time taken was 2 hours, the temperature was 45 degrees and the yield was much higher at 65%. When we compare this data with the conventional method, we see that the time taken was 12 hours. We had to reflux the reaction mixture and the yield obtained was a much lower 45%. After this, let us study about the parameters affecting ultrasonic cavitation. Ultrasonic cavitation is a physical phenomena whose performance depends upon the following parameters, namely frequency, intensity, solvent, temperature, external pressure and lastly the bubbled gas. Next, let us look at some more examples, reactions carried out using ultrasounds. We've already seen one of the Simon-Smith cyclopropanation. We have it on a different substrate and again one can see that the yield is very high, that is 90%. Our next example is of Barbier reaction modified, in which the reaction is carried out for 10 minutes and the yield is 100%. Thirdly, we have a condensation reaction, that is the improved Buchrer-Berg's method, in which traditional method time taken was 4 hours. In the ultrasound, it is reduced to 3.5 hours. The temperature requirements for traditional method were 150 degrees using an autoclave, whereas in the ultrasound, using ultrasound we needed only 45 degrees and the yield changed from 70 to 89%. Next, we have some data on a Refimitsky reaction. Again, one can see here that the yield has improved from a very average 61% to a very high 98%. Our fifth example is of an Ullmann reaction. Normally, it requires a 10-fold excess of copper and 48 hours of reaction time, while in the ultrasound it reduced to a 4-fold excess of copper and just a 10-hour reaction time. That of an oxidation reaction. The oxidation of alcohols by solid potassium permanganate in hexane solvent and benzene is significantly enhanced by sonification in an ultrasonic bath. We have the Kornblum-Russell reaction, in which we can see that the sonification favors a ACT pathway. To summarize what we have learnt in this module: 1. Ultrasound can increase yields, change harsh reaction conditions to milder ones, improve selectivities, and most of all, it can make the reactions that do not proceed under normal conditions occur smoothly. However, sonicated reactions are quite solvent sensitive and these solvent sensitivities are still quite poorly understood. Secondly, ultrasound is gaining significance based on laboratory results and the availability of scale-up systems. Thirdly, we learned that ultrasound increases regioselectivity. Next, we did ultrasound applications that can be envisaged in all types of systems, including homogeneous reactions. Wide acceptance has been gained at a practical and empirical level. However, the theoretical understanding, as we have seen, still lags significantly behind. The usefulness of ultrasound continues to expand into the arena of electrochemistry, photochemistry, and biotechnology. Lastly, ultrasound used in many home appliances and industrial practices.